Hey everyone, we're going to look at incomplete dominance and codominance. These two are very close to one another, but there is a difference, so make sure you pay attention to that. The father of genetics is named Gregor Mendel and worked out a lot of what we've been doing already in class, worked out the rules to genetics and inheritance way back in the 1800s before he even knew about anything like chromosomes and DNA or even genes at the time, but yet was able to work out these rules of inheritance that still work out today. But as we move into modern biology and modern genetics, we found out that his rules work pretty good for a lot of stuff, but not all things. There's exceptions to those rules. There's different types of inheritance. So what we've been talking about so far is what we call Mendelian inheritance or rules of complete dominance, where you have traits that may have more than one allele. You get one allele from each parent, one from mom, one from dad. And when there's two alleles, one can be completely dominant over the other. So we call that one the dominant allele, and the other one is the recessive allele. All right, but we're going to change up those rules. The other rules that we've used so far is we're using capital letters to display the dominant allele, and then its lowercase letter, the same lowercase letter, to display the recessive allele. We're going to change that rule today as well. So again, there's a type of inheritance called incomplete dominance and another one called codominance. These are very closely related, so pay attention. There is a kind of a small difference. So how do we explain this? These are flowers called snapdragons. I used to plant them in my garden when I was a kid. They come in two different varieties, red and white. So if I have a homozygous red plant and a homozygous white plant, and you mix them, well, with Mendel's old complete dominance rules, if I mixed them, I would expect to either get red or white, and whichever one I got would be the dominant one, whether red is dominant or white is dominant. But in mixing the pure breeding snapdragons, we don't get red and we don't get white, we get pink. We get a blend of those two. Very interesting. What's going on here? How about this example? These are types of chickens called erminate chickens. Again, they come in the variety of black and white. And you can go for many generations and see that if you just mate the black ones, all you ever get is black. And you mate the white ones, you only get white. And so you know they're homozygous for their color trait. But then when you mix them and crossbreed the black and the white, you get spotted or kind of this blend of gray. So what is going on there? Because that's not going to fit with our old Mendel complete dominance rules. Here's another example. Type of shorthorn cow. You have white, and then this other one we call it red. I know it's kind of brownish red, but white and red. And so the white ones only ever make white ones and the red ones only ever make red ones. But when you cross them, you get kind of this in-between color and maybe even some spotting, and it's called roan. R-O-A-N, roan. And sometimes there's no spotting. Sometimes they're just completely like a cream color roan. And then finally, these are some flowers that are really neat. Again, I used to plant them as a kid. They're called 4 o'clocks because their buds will open in the morning and close during the evening. I don't know if it's exactly at 4 a.m. and 4 p.m., but they're kind of neat. Again, they come in red and white varieties. The red ones only ever make red ones. White ones only make white ones. But when you cross them, you get pink. All right, so what is going on here? This is what we call, out of the two here that I've talked about, this is called incomplete dominance. This is when neither allele is completely dominant over the other. And so when they get mixed, the traits get blended. You get both of them being expressed at the same time in a blended way, such as red and white making pink, or white and black chickens making kind of a spotted gray, and the red and the white cow making roan. So these problems are done a little bit differently. There's actually two ways to do them. The way that I prefer, this is the way I learned, is that both traits are dominant, so don't they both deserve capital letters? That's what I've always thought, and this is how I was taught. So you actually choose two different capital letters when you do this type of problem. So for instance, for the flowers, I could choose big R is red and big W is white. So we know that the red ones are homozygous for theirs, so they would be big R, and the white ones are homozygous for their trait, so they would be big W, big W. So what do the heterozygotes look like? So when you mix them, here's my Punnett square, big R for the red. Remember, we have to segregate these alleles, so big R at the top of this one, big R at the top of that one. Then bring our W's over here and then fill in the boxes. So all of the boxes will be heterozygous, having a big R and a big W. And so there is the phenotype pink. 100% of them will be pink. So that's how I prefer to do these problems. The chicken problem would be similar, that the black chicken would be maybe big B, and the white chicken, WW. And so then the heterozygotes, BW, would be that spotted gray color. All right, just like with the other problems, does this mean that I've lost the other colors? No, because you can cross a pink flower with a red flower and get a mix there and get your color back. Cross pink with red, you're only going to get pink and red. If you cross pink with white, you only get pink and white, but you can mix things up a little bit. All right, so that's the way I like to do these. And let me switch gears here. All right, so I've got a beautiful Punnett square going here. So I want to do one of these examples for you. So let's say that I am working with those flowers. And so again, my big R is the red allele and the big W is the white allele. So big R is red. Big W is white. It has to be that way because when you have RW, that's when you get pink. All right. So let's say I cross a red flower. So in my Punnett square, I'm going to do this. There's my red flower. Reds are homozygous. Both alleles get a big R. And then let's say I cross it with a pink flower. So pink is always heterozygous. So I need one of each. Does it matter if I write the R or the W first? No, it doesn't. Whatever I decide, I try to do it the same way each time. Now let's fill in the boxes. Big R is always red. Looks like two of the boxes are going to be red. Now fill in this box, big R, big W, and same with this one. Oh, looks like I've got pink. So 50% of my flowers, if I cross a red and a pink flower, 50% of the offspring will be red, RR, and 50% of the offspring will be pink, RW. So that's how those problems work. You just have to choose two different capital letters. After all, both alleles are dominant, and I think they deserve capital letters when that happens. Now I want to show you another way that it could possibly be done. Different textbooks are going to do it different ways. So we use in some textbooks superscripts for the same problem. To me, this makes it a little bit more complicated, but I think this was created to reduce confusion, but I'm not sure if it does or not. So the big C that you see here is for the trait of color. And then they use what are called superscripts for the actual allele. So just like I was doing before, big R, but used as superscripts this time means red, and big W here means white. So they're superscripts. Then we'll transfer down. Law of independence, or excuse me, law of segregation, you can only give one of these to your kids. So this red flower can only contribute the C superscript R, and the white flower contributes the C superscript W. And then here's what pink looks like. And so your pink heterozygote will be C superscript R, C superscript W. That's hard to say. Okay, then here's what a Punnett square would look like. If we cross a pink flower, I just look at the RW. So pink flower, oh, here's another pink flower, RW times RW. So here's my results. Look how when I cross two pink flowers, I get quite a variety. 1/4 of them will be red, RR. Half of them will be pink, RW. And 1/4 white. I can make my garden a beautiful variety of colors that way. So I wanted to show you that there are different ways that you're going to see it done. I prefer maybe my old style way. Maybe I'm just resistant to change. Who knows? But I like this way of doing that. So use whichever you like, and I'll know what you mean, and I won't be confused by it. So that's incomplete dominance. Neither allele is dominant over the other, so you get a blending effect, like red and white making pink. All right. Well, there's this other phenomenon called codominance. It's really similar to incomplete dominance, but there's no blending. So both alleles are still going to be dominant together, but no blending. So a great example of this is with blood type. And blood type is also interesting because there's more than just the two alleles for blood type. You also have three of them. You have allele A, allele B, and allele O. All right, so what is blood type anyway? Blood type is a protein that's on the cell membranes of red blood cells. It's actually a protein with a little sugar tag hanging off of it that is on the outside surface of those red blood cells. And we can call it an antigen because if you put your red blood cells into somebody who's incompatible and has a different blood type, that antigen, that protein, could be spotted by the other person's immune system, and then the immune system could attack it. So for instance, if you have blood type A, your red blood cells look like this picture right here. You have the little A antigens, or the little A proteins on that surface. It's a little protein with a sugar tag hanging off of it. If you have type B blood, your antigen, or your membrane protein, looks a little bit different. So this is a little bit different shape here. You have the B antigen. And so if you have type O, then you have neither one of those. So it turns out with our multiple alleles here, A and B are dominant together. They are codominant. Because if you have the heterozygous condition, where you are blood type AB, your red blood cells will actually display both proteins. They will not be blended. I can look at that blood cell and I can say, oh, there's the A antigen. Oh, there's the B antigen. They're not blended like a red and white making pink. I can distinguish that they're both present separately, or together, I guess you could say. So that's codominance. You don't get the blending effect, but both alleles are definitely dominant, and both alleles will be expressed equally. You will make both proteins. Now, the other circumstance that happens with blood type is type O. O is actually recessive to both of those. So we've got a little codominance going on with the A and B thing, and then we got a little Mendelian complete dominance going on with the O. A and B are completely dominant over O. So you could actually be heterozygous and have A allele from mom and an O allele from dad, and then have type A blood because A is dominant over O. So let's look at what a few problems might look like with blood type problems. All right, so I've switched to a Jamboard here, and on the side, I wrote down the possible genotypes that you might have for the ABO blood groups. There's actually other types of blood groups, but ABO are some of the most important in determining whether you're going to reject a transfusion or not. Your immune system really seems to respond to those A antigens and B antigens. All right, so you might possibly be homozygous A, so that means you got an A allele from mom and an A allele from dad. You might be homozygous B. That means you got a B from mom and a B from dad. And then the codominant condition, you get an A from one parent and a B from another. Then you are AB, meaning you make both of those antigens on your red blood cell surfaces. O, as I said before, is completely recessive to the A and B. So the only way you're going to have type O blood is to get O from both mom and dad. That's me. I got an O from mom and an O from dad. So I am homozygous recessive for my type O blood. All right, but what if you get an A from mom and an O from dad? Well, then you have the heterozygous condition, A and then O. Because I'm doing a codominance problem here, I actually just wrote the O in there instead of like a little A or a little B. It seems to work out easier that way. But I could, if I wanted, use a small case letter for the O's, but I'm going to use just plain old O. It'll all work out in my Punnett squares just fine. And then the heterozygous condition with B, and you get an O from another parent, is BO, right? A B and an O. So here's a popular problem. Let's say that we have a couple, and one of the parents has type A blood. So in my Punnett square, I might write this. And then the other parent, they have type B blood. So I'll write that there for the other parent. And let's say that they have a child with type O blood. Is this possible? Or is someone cheating on someone else? Nope, don't jump to conclusions yet. It's possible because remember, the A and B are dominant over O. So if I write my O over here and my O over there, well, they can definitely have a kid with type O blood. And if I fill in the rest of my squares, here's my AB, my codominant condition. And then BO, so well, that one will have type B blood. And then this one will have type O, or excuse me, A. So literally in this family, they could have kids of all the blood types, type AB, type B, type A, and type O. How interesting would that be? Now, just a little extra information on how the transfusions work. Because type A has those antigens on it, if you try to give type A to someone who has type O blood, type O's immune system is going to recognize those A antigens as foreign proteins, foreign invaders, and will attack it just like it would a virus or a bacteria who's displaying foreign antigens or foreign proteins. That would not be good. That would actually kill a person. Same thing if you give type B or type AB to someone who is O. Again, the O's immune system will recognize those antigens as foreign and will attack those red blood cells, and it could kill the person. How about, can I give A to B? Actually, no. Can I give B to A? No. Can I give A to AB? Yes. Can I give B to AB? Yes. All right. So AB is actually what we call the universal acceptor because they can receive from A, B, and O. Since O doesn't have either of those antigens, then there's no flag, no foreign protein to make the immune system jump into action. So universal acceptor is AB. O is the universal donor because they don't have any weird antigen that is any different in the other types of red blood cells. So again, they are the universal donor and very well sought after during blood drives. I've gotten cards in the mail asking me to come and give my precious O blood during donation time. What's the positive and negative thing? Well, that's another protein that's on the surface of red blood cells. It's a whole different protein. If you're positive, you have it. If you're negative, you don't. So actually, the real true universal donor is O negative because they don't have any of the AB antigens and then they don't have that other positive, it's called Rh factor protein. So if you are O negative, you are really sought after to donate your blood during blood drives to help people out. Now I wanted to show you this, just like in the incomplete dominance, there's different ways that different textbooks and different people are going to do these problems. As you see, I like to just choose the different capital letters. It seems to work easiest for me and be the least confusing. But a lot of other people or other textbooks might use, again, that superscript method. So I is for the gene for blood type. And so if you have a superscript then hooked onto the I, that's the A, and you have type A blood here. Here's type B blood. Then they use a lowercase I for O, type O blood. So they're actually using capital letters, I for the A and B, and then little i's for the O to show that Mendelian complete dominance. And then the superscripts hooked on to the capital I's for A and B. So that's another way that that is shown. So I guess whatever you prefer, whatever works for you, but I just want you to be aware that you're going to probably see it in different ways because different people have different preferences. All right, but I do like how this picture here shows codominance showing both antigens, both the triangle and the circle, that there's not a blend of it, that they're both actually being expressed equally and evenly. All right, so that is incomplete dominance and codominance and some different examples of that. In your worksheet, you might also find another example of a disease called sickle cell anemia that actually displays one of these. So look out for that, and you'll work on your problems and have those done for when we meet in class next time. Make sure you email me or let me know if you have any questions or run into any problems.